Hello, everyone. Good morning or good afternoon, depending on from where you are watching. And I'm very pleased to introduce Anne Churchland, opening this year's virtual ICMNS with the first preliminary lecture. Anne is a professor of neuroscience at the Cold Spring Harbor Laboratory in New York. Her research is centered on understanding neural circuits that support decision-making. Anne's work is truly interdisciplinary. It combines a wide array of experimental techniques to measure and manipulate neural activity in animals while they perform decision-making tasks. And her work also tightly integrates these experiments with computational methods to connect neural activity and behavior. This synergetic approach relies on Anne's interdisciplinary training in her education in mathematics and psychology and further training in system neuroscience. Anne's research was recognized by many awards, including Pew Scholars Award, McKnight Foundation Award, as well as by the Marshall Special Recognition Award for her contributions to promoting women in sciences. I'm very much looking forward to Anne's talk, Discovering Diversity in Decision-Making, Cells, Brains, and Individuals. Welcome, Anne.
Thank you so much for that introduction. I will share my screen now. Okay, so I hope you can all see this. So thank you so much for inviting me and for having me here at this conference. It's a real pleasure and honor to be here. And I really look forward to hearing questions, especially from the students and postdocs in the audience. I find that the people who are newer to the field often have a really interesting perspective. So I may not be able to see your questions in the chat while I'm talking, but I'll see them at the end. And I look forward to a really interesting discussion. So I hope that the work I'm going to tell you about today is relevant to connecting brain and behavior really very broadly. But as many of you know, in my lab, we primarily focus on decision making. And we mostly study decisions that lead to movement. So for example, in this image that you see on the screen, if the mouse makes a decision to eat the blueberry, that will naturally give rise to a movement that will allow him to harvest that blueberry. And that's sort of the first reason that we study decision-making in the context of movements, that it's just a very natural thing to do, and many decisions naturally give rise to movement. And the other reason that we study decisions in the context of movements is that this is a really convenient thing to do from the point of view of a laboratory experiment. So whether you're studying humans pressing a key or animals making a licking movement or a saccadic eye movement, decisions that lead to movement offer a record into the list of choices that the subject made and a number of other associated metrics, such as how long it took to make those choices and so on. But this approach of studying decisions in the context of movements has led to a known challenge. And that is untangling the neural activity for movements from neural activity from other cognitive processes. And this is a challenge that has really been tackled head on by people in the field and have thought about how to disentangle, for instance, a saccadic eye movement used to report a choice from the decision process that preceded it, for instance, by having animals not make the movement or plan the movement until after the decision is over. Another approach is to track the metrics of the movement, such as the animal's head position in an orienting task, and then sort of regress those out when one is looking at the neural activity and wants to understand what component of that neural activity is related to movements and decision versus decision making and so on. And this is all well and good. So in the context of this talk, I'll refer to these types of movements as instructed movements, because we either trained the animal or told the human subject we instructed them to make such movements. And they've been accounted for pretty well by a number of very clever approaches. But what I hope to convince you of today is that these approaches really don't go far enough in terms of thinking of how movements and other cognitive processes interact in cortical activity. So let me formalize that a little bit for you and tell you a bit more about what I mean. So suppose we're studying decision-making in a mouse, and for instance, in the talk that I'll tell you about today, the animal makes a licking movement to report a choice to either one spout or the other. So to just recycle the nomenclature I introduced a moment ago, we would refer to this as an instructed movement. And in this case, it's task aligned in the sense that there's some sort of a stimulus, maybe a flashing shape of some sort. And the instructed movement occurs at a stereotyped time relative to that stimulus because that's what we instructed the animal to do. But as those of you who do animal or even human work know, sometimes animals make uninstructed movements as well. So in this example here, I've schematized a hind paw movement, which is not part of the task. We didn't instruct or train animals to make it. And for this example here, let's suppose that the animal makes 2 hind limb flexions at the time that this flashing stimulus comes on. So we would call that an instructed movement that's task aligned in the sense that there are always 2 of these movements that occur at a stereotyped time in the trial. Of course, these uninstructed movements might have a task independent component as well. That is, they might occur at idiosyncratic moments in time relative to the structure of the task. And then just to complete the matrix, of course, the instructed movements have a slight task independent component, because as many of you in the audience probably know, even when you tell a subject to do things at a particular time, there's a little bit of variation in when exactly that occurs. Okay, so this is kind of the language that I'm going to use in the talk. So what's the big question here? Do brains prioritize cognition and instructed movements, or do other signals dominate? So to get at this, we developed an auditory visual spatial detection task in head-fixed mice, and an auditory or visual stimulus appears on one side or the other, and it's simply the animal's job to report with a licking movement which side the stimulus appeared on by licking to the spout on the corresponding side. This task is a rather simpler version of a large number of rodent decision-making tasks, most of them multisensory, that my lab has developed over the years in parallel in rats and mice and humans as well. And I won't have time to tell you about that today, but there's a published literature on that work. So this is what the task looks like. So at the moment, you're looking at the mouse from underneath. The camera is positioned below the animal. So that's its nose and whiskers at the top of your screen, and its mouth is just below that. You also see 2 lick spouts at the top and 2 handles at the bottom. And the first thing that happens is the handles move in, and the animal makes contact with those handles when it's ready to initiate the trial. Now you're seeing the animal from the opposite point of view, this time from the back, and now you're going to see the stimulus. In this particular trial, it's a visual stimulus, although on interleaved trials, it's an auditory stimulus. And for this trial, it occurs on the right. You can see there's 2 stimulus presentations separated by a short delay. After that, there's a 1-second delay period where the animal has to hold its decision in mind. And then after that, it's time to report the choice. Here's the same view of the camera I showed you originally. And this time the animal's going to make a licking movement to one of the 2 spouts, and the other one will move out of the way. So look at the top of the video because it happens pretty quickly. So he committed to the spout that's on the left side of your screen, and the other one moved away. And that's simply to prevent the animal from changing its mind and ensuring that it will commit to one choice. Okay, so as you can see from the slides I just showed you, we made use of video in this study. We had 2 cameras, one that was on the animal's face and one that was on the animal's body. And this allowed us to track a number of body parts explicitly, such as the pupil diameter, which we estimated and which fluctuated over time as the animal was engaged in the task, not unexpectedly. And we were also able to track the whiskers and the nose, and made some other less supervised use of video as well that I'll tell you about in a moment. This is a similar approach to the one that was used by Carson Stringer in a paper that she published around the same time that we published ours, and made a number of conclusions that I think complement what we observed really well. Okay, so remember our goal here is to determine whether cognitive or movement signals dominate neural activity in experts. So to do that, we wanted to look at cortical activity really, really broadly, because we wanted to be open to the fact that there might be multiple cortical structures that were relevant to understanding how movement and cognitive signals interact. So to do that, we used mice that express a calcium indicator in all of the excitatory neurons. And what that means for people who are not aficionados in the audience is when a neuron fires, it turns green. And that means it emits photons that we can collect with a wide field macroscope. It's a custom setup that we built. And the photons are captured by a PCO edge camera at the top of the setup. And we illuminate the GCamp to get the photons with 470 nanometer wavelength light. And we also have a second hemodynamic channel that we illuminate at a shorter wavelength, and that's to correct for the hemodynamic signals, which are related to, but which are slower and smaller than the direct neural activity that we measure with the GCamp. Slightly more direct, I should say. They're actually both indirect measures. Okay, so this is how we measure neural activity, and there's a few wins to this approach, and to doing it in the mouse in particular, which is why we went this route. So the first thing is this. So this approach allows us to measure neural activity through an intact skull. So we don't have to put a window on the skull, no craniotomy, nothing. We can just see through the skull. We move the skin out of the way, and we can see most of the entire dorsal cortex. Here we also benefit from the fact that mice are lissencephalic, that is, they have a flat cortex with no bumps and grooves. And that puts us in a great position to verify this technique by making maps of visual space. So to do this, we simply show a passively viewing animal, horizontal gratings that are moving vertically and vertical gratings that are moving horizontally. And then we look for each pixel, we say which phase of that cycle does the pixel respond to. And because primary visual cortex and its surrounding areas are arranged retinotopically, we should see a very systematic progression of the phase that each area responds to. And this allows us to identify primary visual cortex and usually around 5 additional secondary visual cortices. So here's the other big win of doing this approach in a mouse through an intact skull, is that we have now both the maps of the visual areas and also the remaining skull landmarks that allow us to align, for instance, the Allen Institute Common Coordinate Frame, so that for parts of the brain that are harder to map using sensory stimuli, we can have a pretty good estimate of where those are as well. So this is the first thing we do in any animal that we have. These are the 11 animals that are in the talk today, and you can see there are really nice retinotopic maps for all of them. This is really handy for aligning the brain, the atlas, right, so that we can tell later on where the areas are, and also verifies that we're getting reasonable signals. Okay, so having established that this technique is giving us reasonable signals, at least in passively viewing animals, it was now time to look at the neural activity during decision making. So what you're seeing here are the same 2 cameras that I told you about a moment ago, the body camera and the face camera. And on the right, you're going to see single trial cortical activity that's associated with the movements that you're going to see on the left. And we'll go through a couple of trials, and I'll just let you kind of look at this activity, which is sort of the first thing that we did when we recorded it. It's slowed down quite a bit, by the way. So as you're looking at this movie, you might notice a couple of things, which we noticed. And for us, there were really 2 things that stood out. And the first one is the activity was quite widespread. And this to us was kind of surprising because this is a relatively simple task. It's just to localize something in space, make a licking movement to that part of the brain. The fact that large swaths of cortex seemed to be recruited was sort of surprising to us. And the other thing that was surprising to us is that the activity was highly variable trial to trial. There was a really quite unique pattern every time the mouse made a decision, even though the decision was ostensibly the same every time. So these 2 features were rather puzzling to us, and we wanted to get to the bottom of that. But before we dug into this single trial, these mysteries of the single trial, as you might call them, we decided to first take a step back and just look at the trial averaged activity to make sure that we're really getting the right signal in the right place at the right time. Because when you see movies like the one I showed you about before with a new technique, their calcium signals, you have to be a little bit skeptical and wonder, well, maybe we're just not really measuring reasonable signals at all. So first thing we did is go to the trial averaged activity to make sure that we're seeing the signals in the right place at the right time. So we took advantage of the fact that in primary visual cortex in area V1, the signals, their temporal dynamics are really well established, really well studied. And so if everything is working according to plan, we should know exactly what we should see. So for instance, when we compare V1 activity for trials where we presented lights versus trials where we presented sounds, those should clearly be very different. So that's what you see here on the bottom. There are the 2 trial types are overlaid here. This is at the beginning before the stimulus comes on. So you can't tell that there are 2 traces here, a red and a black, but there are. And this is the raw fluorescent signal on the vertical axis as a function of time. So when the stimulus comes on, you can see that trials with lights versus trials with tones are very different from each other. And if you look at the gray trace, that's for the lights. I hope you can see that the temporal dynamics are exactly what you would expect. After a short delay, when the stimulus comes on, V1 activity becomes elevated and then slowly peters out when the stimulus eventually goes off. And the 2 traces more or less coalesce around the time of the end of the trial. So this is reassuring. Trial averaged activity in V1 looks exactly like it should. Great. We next compared activity in the left V1 to stimuli presented on the right versus on the left. And of course, as everyone in this audience knows, activity in the left V1 is primarily activated by the contralateral hemifield. That would be stimuli in the right hemifield. So this is a similar comparison to before. You can see when the lights are on the right, we get a much bigger response in V1 compared to when the lights are on the left. And again, the response has exactly the temporal dynamics that we expect. So these analyses demonstrate that at the level of the trial average, we're getting really pretty reasonable signals. This allowed us to go back to the single trial level and try to make sense of these 2 mysteries I told you about before. Why is the activity so widespread and why is it so variable trial to trial? So the goal of this analysis was to take some pixel in the brain, let's say this pixel, and let's suppose this is its activity on trial 1, trial 2, and so on. We wanted to be able to predict and understand and ultimately predict all of these fluctuations. And you can see that the responses on each of these trials are really quite different from each other. And it seemed to me at first to be a rather tall order to be able to predict all of these fluctuations, especially because cortical activity is notoriously variable from trial to trial. But nonetheless, we were undaunted, and we realized that we had a number of good sort of tools at our disposal in terms of behavioral variables that might explain this trial-to-trial variability. For instance, the onset of the visual stimulus, which occurs at a different time on every trial, that's just by design of the experiment. Certainly that would lead to some trial-to-trial variability. The handle grab and the licks occur in different numbers and at different times in every trial. And that's because those are under the animal's control. So of course, they're not always identical every time the animal makes a decision. And finally, the other really important variable that we had was the video. So in addition to tracking some of the movements explicitly, like the pupil diameter and the nose, we also just took the whole video, reduced it down to the top 200 dimensions, and realized that whatever was in the video had the potential to explain the neural activity as well. It was sort of an agnostic way of going in. We said, we don't really know if the video is important, but it might be. So let's consider the possibility that this video might explain some of these single trial fluctuations that we don't understand. So we took all of these variables together and put them in a big design matrix, and then we aimed to find the weights that best map those behavioral variables to the neural activity. And then we tested the ability of the model to predict activity on trials for which we didn't train up the model. I'm going to make a brief technical digression here for people who might have noticed that these regressors are of two rather different categories. So for behavioral events that occur at discrete moments in time, the model learns temporal event kernels. But the other kind of regressors are things that occur rather continuously in time, which we call filterless analog predictors. So let me just tell you a little bit about each of these because they operate somewhat differently in the model. So temporal event kernels are really, really flexible. So even though they're meant to describe the activity related to discrete events in time, they can have really complex temporal dynamics. Like this is a temporal event kernel learned by the model for activity in primary visual cortex. You can see it goes up and down in a rather complex way over the course of the visual stimulus presentation. And these temporal event kernels have been used in other studies in systems neuroscience to understand neural activity related to calcium imaging, and are really borrowed from the field of fMRI, where this technique is used quite frequently. So these differ from the filterless analog regressors. So if you could imagine something like pupil diameter, there is no like pupil diameter event. It's just continuously getting larger and smaller and larger and smaller. So we included these analog signals as well. And under certain circumstances, these analog regressors can have a lot of power, but all they can really do is scale. So if the fluorescence is a scaled version of one of the regressors, then the regressor has a ton of power. But if there's any kind of delay between them, or if there are complex temporal dynamics in the fluorescence signal that are not present in the analog regressor, then those regressors don't really have much of a chance of capturing the neural activity at all. So they really are sort of at a disadvantage in that sense. And I mentioned that because the video regressors that we used, many of them are filterless analog regressors in the sense that there aren't distinct moments in time. The exception being, of course, the video features that we extracted by hand, like the nose and the whiskers. Okay, so that's it for the technical digression. So back to the model now. So I've told you how we set this up and that we aimed to predict those fluctuations on the right on left out trials. So now I'm going to show you how the model did. And you can see for these 3 example trials, that's the predicted response in red overlaid. And you can see it's not bad, actually. We were able to capture a lot of these fluctuations. So I know this crowd is not going to be convinced by a few example trials. So I'm now going to show you how well we did across the entire brain. So this metric that I'll use on this heat map is cross-validated R-squared. So it's how well we were able to predict neural activity on held out trials. And we can do that, of course, for every pixel in the brain. This is collapsed over time, but if we unfold over time, you can see that we're able to predict neural activity both during the stimulus period, during the delay, and so on. So to my mind, this was kind of surprising that we were able to predict so many fluctuations, certainly also encouraging. And this puts us in a good position to say, okay, now let's look under the hood of the model and let's see which of these behavioral features really matters for neural activity. What aspects of what aspects of the behavior are driving these trial-to-trial fluctuations that were so mysterious? So we started off by looking at a group of variables that I'm going to collectively call task variables. And you might ultimately end up disagreeing with the groupings that I've made here, which is fine. They're arbitrary in some sense. So these task variables are, let's say, things that you might include in a traditional decision-making study. So they're things like the onset of the sensory stimulus, the animal's previous choice, previous modality, current choice, whether the previous trial was a success or failure, or so on, things like that. And you can see that these accounted for some amount of variability in the brain. That's great. Here are the instructed movements. You can see that these also accounted for a good amount of neural activity. That's great. Remember, these instructed movements are the ones that have been really well studied by the field. Good thing, because they account for a lot of variability. But here was the surprise. We found that the majority of the single trial variance was accounted for by uninstructed movements, things we never told the animal to do, that as far as we knew really had nothing to do with the task at all. These included movements of the pupil, the hind paw, the nose, the body, whisking movements, the video, and video ME is just the first derivative of the video. It's video motion energy. So this to us was really surprising. So we said, okay, well, wait a second. It can't really be the case that all these uninstructed movements, you know, admitting for the fact that these are movements of very different sorts, right? For instance, pupil probably reflects internal state and so on. But it seems really surprising that these would account for all of the single trial fluctuations in neural activity. And indeed, when we looked a little more closely, we had kind of a hint that something funny was going on. So for example, if you look here, this is the amount of variance explained by the onset of the visual stimulus. And you can see that part of this makes sense. We can explain a lot of variance in the contralateral primary visual cortex, thumbs up. But it was weird that there were these other kind of little places in the brain that seemed to have nothing to do with stimulus onset, where we could explain a lot of activity. That was puzzling. Similarly, when we looked at how well the right handle grab explained neural activity, it seemed to explain it similarly well on the left and right somatomotor areas, which is odd because the contralateral one should have dominated. And finally, this was super weird. We looked at the activity related to nose movements, and although the olfactory bulb, that's at the very top, by the way, explains some of the variance, again, most of the areas that seemed to be most important were these somatomotor regions, which were not obviously related to the nose. So this sort of tipped us off that something was really weird. And we wondered that although we had designed this analysis to include only decorrelated regressors, that maybe there were some correlations that existed despite our best efforts to decorrelate them. So we went back to the model and we decided to do a more rigorous test of the importance of each behavioral regressor. What we did is we went through each one at a time. We kicked it out of the model, we refit the model, we re-predicted all of the trials, we did everything all over again, and asked how much worse did the model do with each regressor left out. So for example, in this case, I've left out the onset of the visual stimulus. We can say how much worse did the model do without that regressor. Well, if it's not important, or if it's related to other, if it's correlated with other variables in the model, then the prediction is not going to be much worse because we don't really need to keep that regressor. There's another one that's sopping up the variance that this one is taking. So we do go through that one by one, and we can then determine what we call the unique explained variance of each of these regressors. Okay, so you remember this from a moment ago. This was the slightly weird contribution of the stimulus onset. When we did this more conservative analysis and estimated the unique contribution of each pixel, you can see that it's very beautifully localized exactly to a primary visual cortex and some secondary areas really only on the contralateral hemifield. Awesome. So then we looked at the right handle grab, and again, the signal really got cleaned up. We see a much stronger signal on the contralateral than the ipsilateral side. And the nose was equally reassuring. When we looked at the unique contribution, we found that just as one would expect, it was completely localized to the olfactory bulb. So we repeated this for all the variables, and one can stare at this for quite a long time. But I think the take-home message is that for most of these movements, the activity was fairly well localized to exactly either the movement effector or the sensory area that you would expect. So there's a bit of a cautionary note here, which is that we hear people say a lot, sometimes related to wide field imaging, to other kinds of neural measurements as well. They say, oh my gosh, when the subject does X, whatever X is, the whole brain lights up. And that's true in our case too, but the only reason that the whole brain appears to light up at the level of the trial average is because there are many different kinds of movements and sensory signals that occur at different times on different trials and can end up being averaged altogether. So when we went back with this more rigorous test, we found that the contributions of the task variables to neural activity although they were appropriately well localized, as you just saw a moment ago, like with the stimulus onset, they were very small. The instructed movements were a little bit bigger, but the uninstructed movements are really the only ones that survived this analysis. So for the remainder of the talk, I'm going to refer to these variables in their groups, the task, instructed, and uninstructed, and I'll just now summarize the contribution of each of these. For the group model, the cross-validated R squared, you can see that uninstructed movements dominated, and for the unique contribution, the more conservative measure, again, the uninstructed movements are completely dominating the neural signal. So this plot here is collapsed over both space and time. I'll just first unfold that for you over time. And that's because you might worry that the uninstructed movements there are there to perhaps support the instructed movements, right? Like maybe when the animal is making the handle grab, it's lurching its body forward, so the two kind of co-occur. But you can see that that's not the case. So the contribution of the uninstructed movements is elevated throughout the entire trial, including the time period before the trial starts. They really dominate throughout the entire trial, and if anything, around the time of instructed movements, like the hold, the handle hold that you can see on the dashed line, if anything, they tend to take a dip at that point because the instructed movements have a bigger contribution. And this is now unfolded over both space and time. This is a movie that shows you the contribution of all of them. You can see the task variables have a brief moment in the sun when the stimulus comes on, but other than that, the uninstructed model is really explaining the majority of the movements. Okay, so I want to point out here that our approach differs from previous approaches, not only in the I guess, number and diversity of the movements that we included in the model, but also for the fact that we allowed for the possibility that any movement in the video might be important for driving neural activity. We just took the video, orthogonalized it to all the other movement parameters in them, and sort of let the model have at it. And the upside of this is it allowed us to make a discovery that we weren't really expecting to make. The movements turned out to be a lot more important than we expected them to be. But the downside of this approach is that we're really left without a convenient sort of low dimensional summary of what actually happened, right? Like, what is it about these videos that are so important in explaining the neural activity? Sort of hard to say. So we can start to get a hint of that. We can start to understand the relationship between the wide field signal and the behavioral video by looking at the weights that are assigned by the model and projecting them back up into the full dimensional space. And that's what I've shown you here. And so each pixel is color coded according to its contribution to the model, the weight it was given by the model. We call this the hot mouse. And you can see it's sort of useful, right? Like there's, you know, pixels around the jaw and the eye that seem to matter, but it really only gives us kind of a hint of what's actually happening in terms of describing behavior in really meaningful language. So understanding what the animal is doing on kind of multiple relevant timescales is something that we're trying to work really hard on now. We started to do that a little bit in conjunction with Liam Panitsky's lab and Scott Linderman in a project that's been led by Ella Batty and a number of others, where we can start to connect different sorts of descriptions of behavior to the neural activity and have a more intuitive sense of what sorts of movements are occurring and in what sequence they're occurring that's driving so many fluctuations in the wide field signal. And I think in the past, I think this new approach is allowing for sort of a bridge that wasn't there before. I think in the past, labs like mine that used what I might call a task-based or psychophysical approach were sort of limited in the sense that we were really restricted to these very small number of metrics, like the animal's choice, its reaction time, and so on, to explain very, very rich neural signals. And that whole approach in the past was really quite separate from what you might call an observational approach, which sort of originally came from ethology, where we would observe, people would observe animals and see what they were doing and try to connect that to animal behavior. One used to have to sort of decide between these two extremes. You were either on the psychophysical side or the observational side. And maybe we kind of don't anymore, right? Now, maybe we can kind of go back and forth between the psychophysical approach where we have really good control over behavior, but still use video to have an observational approach that allows us a much richer characterization. And I think there are a lot of tools in the field that are making that feasible now. Certainly Deep Lab Cut is one of them, where individual effectors are tracked. Other methods like MoSeq that define behavioral motifs and so on. I think in addition, artificial model organisms can help us understand those neural signals as well. And cognitive models can allow us to even further understand the neural signal by estimating quantities that are internal to the animal, such as the amount of accumulated evidence and so on. And I think one approach, one advantage of this approach is it allows us to have a really different view of animal to animal variability. So it used to be that we would study decision-making, for instance, in a rather small number of subjects, and the extent to which those subjects were different from each other was, you might call it a source of frustration, right? You sort of had to take so many subjects that you could average it away, or you would try to do an analysis that sort of minimized that variance. And that was okay, but maybe with these new approaches, we don't have to do that as much. We can, instead of ignoring the fact that subjects are different from each other, we can use that as a way to understand differences in the brain activity that are always observed across subjects. And so this image is meant to schematize that in the sense that these mice here all have the same goal, right? They're all trying to get the cheese, but they're employing very different strategies in the service of that goal. And I included the artificial mouse to convey the idea that we might learn about the strategies of artificial organisms in a way that could be helpful in interpreting brain activity as well. And this idea is sort of fleshed out in a review that I wrote with Anna Arai, who's a postdoc in my lab, Simon Musal, who's one of the leaders on the Y-Field project, and also David Susillo. Okay, so to start to get a sense of how this relationship between task and movement variables to try to understand it better, right? While we're still in the early stages of that new methodology I just described, we thought, well, let's image activity as animals are learning the task. And that will start to give us some insight on how these different variable groups interact. So the first thing we did is look at the contribution, the CDR squared, as a function of training days of each of these three variable groups that I've been telling you about. So you can see a few things happen here. So first of all, the uninstructed movements always dominate the neural activity. But their contribution goes down slightly. Task and instructed movements tend to go up. That's sort of expected because originally the animal doesn't know much about the task or its structure, and it only becomes meaningful over time. But we wanted to understand this slight decrease in the impact of uninstructed movements. And we had our first idea is that maybe the impact of individual uninstructed movements changes over the course of the animal's training. So in other words, maybe a particular movement, say moving a forepaw, has a bigger impact early in training compared to late in training. So to get at that, we simply looked at the weights that were assigned to the uninstructed movements as a function of training. And that's what I've shown you here, the normalized beta weights on the vertical axis as a function of the mouse's training days on the horizontal axis. And you can see like not much really happened here. So it seems like whether the animal makes a particular movement early in training or late in training, it has pretty much the same impact on the neural activity. So it's not the case that the impact of individual uninstructed movements changes. So then we had another idea. We said, well, maybe what's changing is the frequency and timing of uninstructed movements changes. And that turned out to be the case. So let me be really concrete here. So let's think about whisking. So this is not an interesting plot. This shows you the rate of whisking as a function of time in the trial. So remember, 2 seconds is when the visual stimulus comes on. And you can see that the animal early in training is pretty much equally likely to whisk at any moment in the trial. So it's completely task independent, to use the language that I introduced at the beginning of the talk. When we look at whisking late in training, it's a totally different picture. You can see it becomes highly structured. The animal has a very stereotyped whisk right at 2 seconds, and then another slightly, that's when the visual stimulus comes on, or auditory stimulus, and then a slightly less stereotyped one at about 4 seconds. So remember, whisking is not part of the task. We did not train the animal to whisk, and yet this whisking, clearly from the point of view of the animal, is something that it does in a very stereotyped way that differs dramatically from experts versus novices. So to say this in a formal way, we found that the task-aligned contribution of these uninstructed movements it grew dramatically over training days, but that the task-independent component tended to go down. So I want to just give you an intuition for what these words really mean: task-aligned, task-independent. What is that? And should we think about these as occurring in humans in any kind of meaningful way at all? And we're still thinking about that deeply, but just let me give you an intuition here. So an example of a task-aligned uninstructed movement would be a repetitive movement that a well-trained expert does in a reliable sequence in advance of a learned movement. And an example of this is baseball players. So this is Nomar Garcia-Pera, who plays for the Boston Red Sox. And you can see he has an elaborate series of movements he makes that are not, to us, obviously related to hitting the ball. Right? He's pulling at his sleeves, his gloves, and so on. These are really common. Here's another one. This is Mike Hargrove, who has a really nice fidget. And these fidgets are interesting. They probably, for the point of view of the learned expert, are very much part of doing that trained movement, although to those of us observing, it's not obvious how they are related. They've been reported by other people in both humans and in animal work by Ben Olszewski and others. The other kind of movement, which I almost find a little more interesting, are these task-independent, uninstructed movements. Remember, these are things that don't occur in a stereotyped way relative to trial events. And I think that actually humans really do those things. Leg rocking, leg shaking, chair rocking, head scratching, eyeglass stuff. The lower one is me playing with my Fitbit. Humans do these things a lot. If you, next time you're in a seminar, which hopefully won't be too far in the future, if you look around, people are moving all the time. And one intriguing possibility is that under some circumstances, these task-independent, uninstructed movements are a signature of trying to focus on a cognitive task. And so it may be that although we tend to find them annoying in both humans and possibly in animals as well, that they're serving some interesting function of cognition that has yet to be understood. Okay, so I want to stop on time, so I won't have time to go through in detail the potential concerns, but naturally we worried that these results are unique to wide field imaging, only true in the cortex, or only true with calcium signals. So we then repeated all these results with 2-photon imaging, where we looked at single cell neural activity. We measured signals from a large number of neurons using exactly the same method that I told you about before. We had about 12,000 neurons altogether, and again, found that the uninstructed movements totally dominated the neural signal. We also worried that these results were due to the fact that we were measuring calcium signals. Calcium signals are not spikes, and they have a complicated relationship to true neural activity. So we also repeated these experiments with Neuropixels probes in an animal doing a slightly different task, and again, found the same conclusion, which is that movement signals dominate both in subcortical structures and in cortical structures, and it doesn't matter whether you're doing calcium imaging or measuring spikes. So in future work I won't have time to tell you about, we're starting to look at some of these signals in specific populations of excitatory neurons and are finding some really interesting differences across these populations. Okay, so I've told you so far that cognitive experts engage in diverse uninstructed movements with profound consequences for neural activity. That robust movement representations are widespread in both cortical and subcortical structures, and that learning introduces dramatic changes into the timing and frequency of these movements. In our current work, we're trying to better understand the function of these, their prevalence in different classes of pyramidal excitatory neurons, and their relationship to multiple levels of behavioral description. So I'll stop there. The leaders on this project were Simon Niesal, who just left my lab to be a PI in Germany, and Matt Kaufman, who's now a professor at the University of Chicago. And these are the people that supported our work, and we're really grateful to them, and also grateful to you all for listening, and I would be happy to take questions.
Thank you very much for a beautiful talk, Anne.
No
and
problem.
we have a series of questions which came up in Q&A, and I will just read those questions to you.
Okay, so I'll stop share. I guess I can always reshare if I need to. Right.
So the first question, I think you already partially answered that question, but it got the most votes, so I would voice it again. How long were the mice trained for, and what happens over training? So the question was how the structure, how unstructured movement, whether uninstructed movements remain the same or become weaker or get stronger over the course of training. And I guess you partially answered that, but interesting question is about relationship of those movements to neural activity. If you maybe would like to address this again.
Yeah, sure. I'll share that slide again because I went through that quickly. So yeah, we trained the animals for, it depends a little bit how quickly they learn. So it's not always clear what would count as the training time. But here you can see data that's collected over about 45 days. So pretty long time. So the animals are trained about 5 days a week. So that's about 9 weeks of training. And then we looked at the contribution of each of the variable groups separately. The uninstructed is in black, the instructed is in blue, and the task is in green. So if you look at, for example, just the task variables, or initially the task variables have very little impact on neural activity, but that grows over time. And I think the intuition for that is that a lot of these stimuli don't really have meaning to the animals initially, and also they aren't really making the instructed movements with much frequency or precision because they're just figuring it out. It was the decrease in uninstructed movements that was more mysterious. And that decrease, the fact that that black line goes down over time, was what drove us to do the analysis, where we first looked at the beta weights assigned by the model to the movements and found that they did not change. And then looked at the frequency of and timing of the movements and found that they really changed quite a lot.
Okay, thank you. Another question which comes from Dmitry Todorov is how do contributions of task, instructed and uninstructed movements change over brain areas?
Yeah, okay, so the right slide for that is, well, there's a few slides I can show. Let's see. Yeah, let's do this one. Just a sec. Okay. So the key one for this is probably this movie here. So this shows the contribution of each of these three variable groups over time in the trial. I'll show you this movie twice because on the first time I'll tell you what it is, and then the second time you could actually look at it. So each of these shows the whole brain map of each of these three variable groups, and it's fluctuating over time because of course the contribution of those also fluctuates over time. So for instance, the contribution of the task variable group is really dominated by the onset of the visual stimulus. That's mostly because V1 is big and in the back of the brain it has a spatial map to it. So the contribution of that variable group is clear and transient only when the visual stimulus is on. Instructed is in the middle, and uninstructed is just one to the right of that, and then the full model is one over. So you can see that that uninstructed movements explain neural activity really all over the brain, including primary sensory areas like V1. But the task variables and instructed movements are more localized to the stimulus or effector at hand. So I'll show you that movie just one more time so you can look at it. There we go. Okay, so this is the baseline period. Animal does a handle grab in a second. You can see that the somatomotor areas start to get active. Stimulus comes on. V1 is transiently relevant. Then there's a delay. And then around the time of the movement, then there's more activity again. Does that answer the question?
Yes, thank you. That's great. So another question comes from Laura Mediavilla, and she thanks you for the beautiful talk in the first place. And she also wants to know how this analysis could be possibly applied to spontaneous activity where there is no behavioral task.
Yes, it definitely can. So that's actually what we did in the Neuropixels experiment. So let me just go back to that for a moment. So in this experiment, it's sort of a long story, but the part of the experimental data that we analyzed was when the animal was head fixed and was freely moving. Sorry, was head fixed and could make spontaneous movements, but was not engaged in any kind of a task. It was just passively viewing visual stimuli that were presented. So because there were visual stimuli presented, we had a task model. Those are just the visual stimuli. That's basically just the stimulus model. There are no instructed movements in that case because the animal's not in a task. But other than that, it's exactly the same analysis. So you just don't include instructed movements because there are none. So all of the movements are uninstructed. So you can definitely do this analysis in animals that are not engaged in the task. It's just exactly the same, except there's no instructed movements. That's a little bit related to what Carson Stringer did in her 2019 paper. The animals were passively viewing and not engaged in a task, and they were looking primarily in V1. I think, to my mind, there have been a number of papers that have demonstrated that when animals are not engaged in a task, that there's a lot of fluctuations in brain activity that are due to kind of changes in state or movements, at least like movements like running and pupil diameter and so on. And I think for us, and maybe for a lot of other people, we sort of had the sense, or at least the gut feeling, that was really restricted to animals that were passively viewing. And we felt like, well, in our case, our animals, like, they are decision-making machines. Like, they don't have brain states fluctuating all over the place. They initiate a task deliberately by making that contact. So we really expected that in these well-trained experts, the cognitive signals would dominate in a way that they did not in passively viewing animals. But I think what we discovered, and this to us was really surprising, that our view of the animals as kind of these decision-making automatons was really way off. Like, yeah, they're making decisions, they're experts, they're really good, but they also have a lot going on that we were not paying attention to before. And I think this kind of highlights my enthusiasm for this idea of bringing together the psychophysical approach where animals are well-trained and execute many, many movements, but are also, you know, kind of doing their own thing in a way that we can start to capture with video and with estimates of internal state and estimates of cognitive variables and so on.
Thank you. So related to Neuropixels, there are two more questions about these Neuropixels recordings. One is from Dmitry Todorov, who wants to know if you were able to record from subcortical structures during these experiments.
Yes, so actually in this slide here, VC stands for visual cortex, SC is the superior colliculus, and MRN is the midbrain reticular nucleus. And you can see that in all three areas, we found that the movement signals really dominated. So in this case, we were only looking at uninstructed movements, so we aren't able to compare instructed versus uninstructed, but we know that even in these subcortical structures, the movement signals are very, very large.
And another question about Neuropixel is whether you were able to look in different cell types, for example, excitatory versus inhibitory neurons, and what was their contribution. And when you analyze this data, were you looking in mean firing rates of these single units?
Okay, well, let me answer those separately. So we're never looking at mean firing rates, right? Because we're always trying to, the model is by definition, it's a single trial model. So we're always trying to predict the single trial activity. We can, if you look at this slide here, the mean activity is on the left. You can predict the mean activity really easily. We have lots of, that's very feasible. It's predicting the single trial fluctuations that's really difficult. And that's what we did in both the calcium signals and also in the electrophysiology that you see here. So on the right, that's because it's single neuron, you get a neuron by neuron breakdown. So you can see for each neuron, how much movement contributes, stimulus contributes, and so on. And you can see it's kind of idiosyncratic. Like this neuron here, the stimulus really dominates, right? And this is not a movement dominant neuron. In this one, the two make equal contributions. So this neuron is driven both by stimulus and by movement. And in this neuron over here, the movement really dominates. So individual neurons, somewhat surprisingly, reflect rather idiosyncratic combinations of these different types of variables. And I think that has sort of implications for how correlated you would expect them to be and so on. In terms of looking at different cell types, yeah, we're super psyched about looking at different cell types. We have another paper that looks at excitatory versus inhibitory neurons during decision making. For this study, it's a little easier to do the cell type stuff with imaging, actually, right? Because you can just make the cell type, you can distinguish the cell types by color, by wavelength, which is a lot easier than doing it with electrophysiology, where you have to do optotagging, which is a complicated thing. But we were really fortunate during a collaboration with Josh Wong, who's my colleague here at Cold Spring Harbor, to get access to some mouse lines that targeted specific excitatory cell types that are defined by developmental lineage. We studied FESF2 cell types that project, we call it corticofugally, so to the superior colliculus and the brainstem, and plexin neurons that project corticocortically and also to the striatum. And this is a really, really different way of looking at neural activity. And we find that especially, we find really interesting differences between these two. This is sort of a subject of a different talk, and this work is still kind of developing. But you can see that, for example, for the task variables, they're much more localized in the FESF animals and much more distributed in the plexin animals. And I think this approach really highlights the big win of wide field imaging. So what I told you about today, looking at all excitatory neurons together, you're getting a lot of different cell types really mixed together in a way that's just kind of a loss relative to like 2-photon imaging, except for the scale. But here we can look at the specific contributions of distinct cell types and learn about how those different cell types work together within and across areas to support decision making. Great.
I have one more question from Jonathan Rubin. And the question is, it looks like contributions of uninstructed, instructed, and task-related movements all explain less variance as training progresses. So what takes their place?
Yeah, so could you just repeat the very last part of that question? I didn't hear the
last part is what takes their place?
Yeah, so we were able to explain overall about 35 or 40% of single trial fluctuations. So that's a lot. Like usually when you do an electrophysiology study, the Fano factor is like 1 or sometimes even higher than 1, right? Like your variance is equal to the mean. So we explained really a lot of the fluctuations, but of course you're totally correct. There's still, you know, the majority of the variance we still can't explain. So I don't of course know the answer. I have a couple of ideas. So one possibility is that there are more signals, maybe they're movement signals, maybe they're other kinds of signals that we didn't measure that also account for those fluctuations. So we didn't, for example, measure respiration or heart rate. Maybe those things account for some of the fluctuations. There are aspects of internal state that we were only able to approximate, like with pupil diameter, but could have estimated with more precision that might have accounted for more of the variance. So that's sort of the first class of possibility, that there's other variables. And if we put those variables in, we'd eventually explain all of the single trial fluctuations. The other possibility is that some of the fluctuations do reflect true stochasticity. And certainly there's a long history of work suggesting that stochasticity in the brain is inherent to neural circuits and may even serve some function and so on. So I can't distinguish those two hypotheses at the moment, but it's going to be really interesting to try to dig deeper on that.
Okay, thank you. So I would like to ask one methodological question, which arose very early in the talk from Tom Burns. And he was asking what the advantages of wide field calcium imaging relative to fMRI?
Oh yeah, so great question. So the spatial and temporal resolution, I guess there's really two reasons. So the first one is that the spatial and temporal resolution are vastly superior. So we sample at we sample at 60 hertz, but we divide it in two because we have the hemodynamic component. But it's a much faster rate. Remember, fMRI is ultimately measuring hemodynamics. We are measuring calcium signals, which are a much more direct proxy for neural activity, and we can catch those photons directly with a camera. So our spatial and temporal resolution is much better. Having said that, I think it's a totally fair question, right? Because fMRI, you're studying it in humans. That's the big win. But I think that the other major advantage, and that makes wide field imaging a really important technique, it's the cell type specificity. So when you do fMRI, you can only ever look at the pooled contribution of very different cell types. In some cases it might be fine to pool those cell types together. In other cases, it absolutely isn't. There are different classes of excitatory neurons that differ completely in terms of their developmental lineage, probably also in their function. And if you can look at those cell classes separately, you'll get a very different picture of brain activity.
Oh, that's fascinating. Okay, thank you and so much again for answering all these questions. We will have to close this session to stay on time, but I invite everyone to join our Slack channel. And I hope, and you would be able, there is a special channel dedicated to your presentation. And if you have more questions, I encourage everyone to text Anne over Slack, and I hope you would be able to answer some of those questions.
Yes. Okay. I don't know where the Slack is, but I will find it, or someone can tell me.
Yeah, we will help you to identify Slack. And everyone else, you can look in the program to find instructions on how to join the Slack channel. So the next session will start shortly. And for the next session, we will split in 2 separate channels. So just follow the program to find which channel you find more interesting, and see you all soon shortly.